To start out, I just want to recognize the work that went into setting up this series, and in particular, a few individuals who played a particularly important role here. First, the former IPN director, Joe Rochford, who was instrumental in creating this initial series in interactions with Bernice Grafstein. Um, Joe is retired, but he's still very much in our thoughts. The Neurodevelopment Office has played an important role in administering the gift from Dr. Grafstein, in particular, Ms. Sarah Slack. And of course, Adriana Scambetera, who is the IPN project manager, who really was the person who brought all of the elements together today to make the event happen. So thank you, Adriana. And of course, we've had assistance from Debbie Warshawski and Marc-Andre Meloche in setting up the initial registration and getting advice on how to set up the cocktail hour. So I want to tell you a bit about Dr. Grafstein, those of you who don't know. Bernice Grafstein is truly a kind of McGill legacy to neuroscience. She's currently the professor of biology and biophysics at the Weill Cornell School of Medicine in New York. She's the Vincent and Brooke Astor Distinguished Professor in Neuroscience. She's here, I think. Dr. Grafstein received her BA in physiology from the University of Toronto and then did her PhD here at McGill University in the Department of Physiology, working with Benedict Delael-Burns. And during her time here at McGill, she actually did really important work on the phenomenon of spreading depression and helped us to understand this quite important phenomenon in epilepsy and in cortical function. Bernice then went on to do brief work at the Rockefeller University. She also worked briefly with Victor Hamburger at Washington University, where she did quite impressive work on nerve regeneration, and then finally joined the faculty of the Weill Cornell Medical School in 1969, where she's been ever since and continues to teach and to work. Bernice, as I said, did work to characterize the phenomenon of spreading depression. She studied neuronal connectivity under Victor Hamburger at WashU. Some of the important work on regeneration in the goldfish optic nerve that she did, and the discovery of intracellular and transneuronal transport of proteins in neurons. So all very seminal work. But equally important has been Bernice's contribution to the neuroscience community. She was Society for Neuroscience president in 1985, the first woman president of the society. And she subsequently received the Mika Salpeter Lifetime Achievement Award from the society in 2003. Bernice was elected a member of the American Academy of Arts and Sciences in 2021, and is a life trustee of the Grass Foundation. She's received many other prizes. And Bernice has held a very, very high position, an important position in her interactions and in her gifts for science education and science outreach. And in particular, she gave the gift that allows us to have this annual series with the hope of both introducing young neuroscientists to really exciting, first-rate work in the field, also bringing together the Anglophone and Francophone communities in neuroscience in Montreal. And I think you'll find that today's speaker definitely accomplishes the main goal of being an extraordinary scientist and speaker. So to introduce today's speaker, we have Abigail Ralph, who's an MSc student in the lab of Joanne Stratton at the IPN, and I'll hand over the podium to Abigail.
Hi, everyone. I'm really excited to be introducing Dr. Catherine Dulac. A few students and myself had a great lunch with her, so it's going to be a great talk. Why are we wired to connect with each other? What impacts do loneliness and isolation have on social behaviors? What are the neurobiological underpinnings of social desires? Dr. Catherine Dulac is deeply inspired by these questions and has built a successful career in pursuit of this knowledge. Catherine Dulac is an inspiring French-American molecular biologist and neuroscientist renowned for her groundbreaking work at Harvard University. As of 2022, she holds a prestigious Samuel W. Morse Professorship, an honor Howard University bestows upon its faculty who has achieved greatness across departments. Her journey to this pinnacle of to this pinnacle of academic excellence began with her roles as the Higgins Professor of Molecular and Cellular Biology and the Lee Espelda Professor of Arts and Science. Dulac's story exemplifies the power of determination, scientific curiosity, and the relentless pursuit of knowledge, making her a true trailblazer in the field of neuroscience. Catherine Dulac grew up in Montpellier, France, with parents who were academics in the humanities. In 1982, she entered the vaunted École Normale Supérieure, graduating with a Bachelor of Science 4 years later. Dulac completed her PhD research under Dr. Nicole Dunienne at the Institute of Molecular and Cellular Embryology, affiliated with the College of France and the French National Center for Scientific Research. She defended her PhD at the Pierre and Marie Curie University in 1991 in Paris. After completing her PhD, Catherine Dulac became a postdoctoral fellow at Columbia University in Dr. Axel's group, motivated by her research and willingness to work with mice as model organisms. In 1996, she joined Harvard University's Department of Molecular and Cellular Biology, becoming a full professor by 2001 and serving as department chair from 2007 to 2013. She's also affiliated with Harvard University's Brain Science Initiative and other research centers. Dr. Catherine Dulac's research has explored olfactory signaling and the neuroscience of sex differences and parenting behaviors. She was the first to identify genes encoding pheromone receptors in mammals, specifically veronasal receptors. Her work on the TRPC2 ion channels in mice revealed its importance in sexual behavior differentiation, with female mice displaying male-like behaviors when lacking this gene. She also studied neuroregulation of parenting through gland expression neurons and has investigated sex-specific differences in the amygdala. Catherine Dulac's career is a powerful example of excellence and perseverance. She has received some of the highest honors in science, including the Breakthrough Prize in Life Science in 2001, and also being elected to prestigious institutions in science. For example, the National Academy of Science in 2015 and the French Academy of Science in 2007. Elevated to the Office of National Order and the Legion of Honor in Neuroscience in 2022, Dulac's achievements reflect her extraordinary contributions to the field of neuroscience, inspiring others to pursue their passion with determination and with brilliance. On behalf of McGill University and the Integrated Program in Neuroscience, please help me give a warm welcome to Dr. Catherine Dulac.
Thanks, Abigail, for the beautiful and very nice introduction. Thanks, Ed, for the invitation to give this lecture. And thank you also for Dr. Grafstein for the donation that led to this invitation. So as you've heard from Abigail, my lab is focused on circuit underlying social behavior. So why is that? Well, for a very simple reason, which is that social interaction is really, or social stimuli are really some of the most salient stimuli in animals' life, in humans' life. When animals have choice between cocaine and a social conspecific, they will choose the conspecific. There's something very special about interacting with other animals of your own species. And overall, as human, we know how much we enjoy being together and gather in large audiences, whether it's for talk, for concert, for a party. We also spend a lot of our mental time thinking about the people we know, the people we love, the people we hate. So social interaction really is something very profound, very important for our life. And similarly, the inability to perform or to engage into social interaction is one of the most debilitating aspects of mental illnesses. And to these days, we don't really know why that is the case. And so early on in my career, I became really fascinated with the idea of trying to uncover the specific neuronal population in the brain and their interconnection, the circuit that underlie social interaction. And for this, we've been using any type of approach that were necessary, from very molecular, cellular, systems level, and behavioral approaches to try to uncover the logic of the function and the nature of circuitry underlying social interaction. And today I would like to tell you about two topics that started in my lab about 6 to 8 years ago. And one is social isolation and the other one is sickness. And I mentioned we started this well before the pandemic, and obviously they became very interesting in the context of the recent pandemic that we experienced. And I'll tell you also the context, how we came to think about these issues and talk about them. So overall, just to introduce the way I've been thinking about circuits of social behavior, we really are trying to understand circuits of social behavior in a brain-wide manner, starting by stimuli that are detected that indicate an animal of the presence of a conspecific circuitry that are involved in the processing of these sensory signal, central circuitry that are state dependent, whether it's depending on sex or the physiological state of the animal, and then the actuator of social behaviors. And early on in my career, as nicely summarized by Abigail, we were interested in the stimuli that trigger social behavior, particularly a signal identified by the voronasal organ, and using mutation in the TRPC2 ion channel that is essential for VNO signal transduction, we were able to identify the rules underlying the sex specificity of social behavior. This, in turn, led us to investigate more central questions on circuit of social behavior, and particularly on what we really picked as naturalistic paradigm to deconstruct social behavior. So not only one particular social behavior, but actually the choice sometimes between different and opposite type of social behavior. And one really great example, I think, is parent-infant interaction, in which animal can be either parenting, for example, this mother chimpanzee here, or infanticidal, for example, this langur here with the cadaver of a little infant that it had killed. And we identify specific neuronal population that govern this behavior. For example, these galanin neurons in the preoptic area that are essential for parenting behavior, both in males and females, and these urocortin 3 positive cells in the perifornical area that are essential for infanticidal behavior, both in males and in females. And so these, in turn, led us to wonder what are the rules underlying state-dependent behavior? You know, animals do not display the same behavior always. It depends on their physiological and other state. And so this led us to wonder about sickness behavior, how sickness influences social behavior, and as well as the presence or the absence of other conspecifics. So let me start with sickness behavior. We have all experienced at some point in our life what sickness does to behavior, and we know that it affects a really large range of behavior, from shivering and warm seeking, so going under a blanket or drinking hot soup or hot tea, to fever, so elevation of temperature, but also loss of appetite, loss of energy, this lethargic state of being sick, social avoidance, increased pain sensitivity, etc. So there are a lot of behaviors that are affected when an individual is sick, has an infection, whether it's a bacterial or viral infection. Now, interestingly, all this range of behaviors that are called sickness behavior as a whole are shared with other animals. And this has been tested in mammals, for example, in rodents. For some reason, people test birds and particularly pigeons, but also cold-blooded animals, insects, lizards, crickets, all display almost exactly the same range of sickness behaviors. Now, obviously, cold-blooded animals cannot have fever because they cannot change their body temperature, but what they do is they go to a warmer place in order for their body temperature to go up. Now, until I actually worked in that topic, I assumed that all these awful feeling, all these awful sickness behaviors were due to either the toxicity of the pathogen or the immune system being in overdrive. But in fact, an idea that has emerged recently is that all these behaviors are actually adaptive. They are there to help the organism better survive pathogens. For some of them, it's very obvious, warm seeking and fever, so elevating temperature is actually good to inhibit the multiplication of the pathogen and also boost the function of the immune system. Lethargy, you try to save energy to fight the pathogen. Social avoidance, you try to avoid to spread the disease, etc. Some are a little less obvious. For example, loss of appetite. It would seem counterintuitive. If you're sick, you should actually eat and have more energy to fight the pathogen. It turns out that this is not the case, and that an immunologist at Yale, Ruslan Medzikov, injected animals with either viruses or bacteria and found that when he gavaged the mice, they all died compared to the control that display a loss of appetite. So in other words, for a rather complex metabolic reason, loss of appetite is actually also adaptive. So where are all these adaptive behaviors coming from? What is coordinating all these behavioral changes? And the pioneering work of Cliff Saper at Harvard Medical School proposed that there are specific centers in the brain that organize and coordinate some of these changes. And in particular, in the case of fever and change of body temperature, he showed that pathogen trigger the innate immune system that leads to the release of cytokine. And this cytokine interact with this particular area of the blood-brain barrier called the CVO, or circumventricular organ. This is an area of the blood-brain barrier that is fenestrated such that molecules can actually pass the blood-brain barrier, and in particular amplify prostaglandin signaling, either going through the CVO or being secreted by the endothelial cells of the CVO and by microglia. These prostaglandin in turn inhibit, through the EP3 receptor, inhibit the function of this particular neuronal type, these warm sensing neurons that were discovered by Zach Knight a few years ago. These neurons are essential for temperature control in the body. And basically, if you don't inhibit the function of these neurons, the temperature cannot go up. So you cannot have fever if these neurons are not inhibited. So the way then fever works is that this peripheral immune response lead to prostaglandin inhibition of these warm sensing neurons, and in turn now thermogenetic center can work and lead to a rise of temperature. And so what we wonder is that what about not only fever, but the other symptoms of sickness? How are they controlled by the brain? Can we identify the corresponding circuit? And this work was done by Jessica Osterhout, who now has her own lab at the University of Utah. And the first thing she did was to optimize an experimental induction of fever and also other sickness symptoms in mice. So we cannot inject bacteria, we cannot inject viruses in our animal facility, but the way people typically do in the field is to inject molecules that mimic a bacterial or viral infection, in particular lipopolysaccharide S, so LPS or poly-IC, mimic respectively a bacterial infection and a viral infection. And when you inject a mouse, you can see that about 2 hours later, there is a peak in temperature that reflects the induction of fever, and then it goes down. And when she tested other type of behaviors, for example, temperature preference by putting the mouse on a gradient of temperature, chow consumption, or locomotion, she found that both LPS and poly-SC injection induce an increase in temperature, a decrease in food consumption, and a decrease in locomotion. So overall, although these are just molecules and not the actual pathogen, they very nicely mimic not only fever, but also other sickness symptoms. So what are creating and orchestrating these various symptoms? And so for these, we decided to use a strategy that we had used multiple times in the lab, which is to inject LPS and then identify activated neurons based on the expression of CFOS or FOS, the immediate early gene that reflect acute neuronal activation. And as you can see, we found this really interesting pattern of CFOS positive neurons in this area here at the bottom of the brain, bottom of the hypothalamus, in an area called the ventral medial preoptic area, or VMPO. You can see in black the CFOS positive cells also along the ventricle. Now, this is a very interesting location because it's right above the CVO, this fenestrated broad-brain barrier that I mentioned to you earlier. Now, there are other CFOS positive cells after LPS injection, but these really triggered our attention for its particular position. I should also say that these neurons are inhibitory neurons, which will be important to understand how they function. So I'm going to tell you a little bit later what the identity of these neurons are, but in the meantime, we use the trap system that was developed by Li-Chun Luo's lab in order to perturb the specific population that was activated after LPS injection. And so this is called the TRAP2 system. It's basically the CFOS promoter driving an inducible Cre-ER. And so when this mouse is now crossed to either a conditional reporter mouse or conditional actuator mouse, these enable, after activity, for example, LPS injection and tamoxifen injection that induce the Cre-ER, to express specifically either D-tomato, so reporter, or an actuator, either excitatory or inhibitory, optogenetic or chemogenetic actuator, to perturb the function of the cells. And in control, we are able to perfectly recapitulate the expression of the reporter in the CFOS positive cells. And now we were able to either activate these neurons or inhibit their function and see what the impact is on animal behavior. So to activate them, we use this LPS trap system. So we trap the neurons with LPS injection and tamoxifen. So now all the LPS activated neurons express the Cre recombinase, and we inject into that mouse this flex virus expressing an excitatory dread. So this is a receptor sensitive to artificial ligand that enable specifically those neurons to be activated. And then to perform loss of function, we now, in LPS trap mice, inject a flex diphtheria toxin virus. So this enables us to kill these neurons. And then we can inject LPS and see whether there's any effect, any difference between the control and the trap mouse. So when we perform this LPS trap mouse and inject this excitatory dread and activate that receptor with CNO, we see that the animals see an increase in temperature. When we kill these neurons and inject LPS, we see that there's no longer any LPS-induced fever, suggesting that these VMPO LPS population are essential for fever. What about the other sickness symptoms? Well, we find that when we activate these neurons in a perfectly healthy mouse, we see an increase in preferred temperature, a decrease in food consumption, no effect on locomotion. When we kill the VMPO-LPS neurons and now treat the animal with LPS, we see no longer any LPS-induced worm-seeking. We see still a reduction in food consumption, and we also still see a reduction in locomotion. So these suggest that these neurons are required for worm-seeking. They participate, but they are not essential for loss of appetite, and then they have no role in reduction of locomotion. So how do they fulfill this function? Well, we look at the projection of these neurons, again, using the trap mouse and injecting this flex virus that express not only a fluorescent reporter GFP, but also synaptophysin MRUBY fusion protein that labels specifically presynaptic terminals. And we found many, many brain areas that receive input from the VMPO-LPS neurons. Two of them really triggered our interest. One is the MNPO. This is the area that has these worm sensing neurons that I mentioned in the introduction. And the other one is the arcuate nucleus, which is an interesting area now to regulate food consumption. So how are these VMPO, do these VMPO-LPS neurons modulate the function of the MNPO and arcuate nucleus? What we found using rabies viral tracing is that the VMPO-LPS neuron send direct projection to MNPO neurons expressing the marker ADCYAP SNCG, which are the marker of worm sensing neurons. And again, remember, these VMPO-LPS neurons are inhibitory. Therefore, this might be the mean by which the animal can develop a fever. And indeed, when we manipulate the projection of VMPO-LPS neurons to the MNPO, we see an increase in body temperature and absolutely no effect on food consumption or any other behavior. So we think that this particular node is responsible for fever, for the development of fever during an infection. Then we looked at the projection to the arcuate nucleus and found, again, using viral tracing, that the neurons that receive direct input from VMPO-LPS neurons are neurons expressing the neuropeptide AGRP. These neurons are known to be responsible for appetite control. And so again, during excitation of the VMPO-LPS neurons, these neurons are inhibitory and therefore are likely to inhibit food consumption. And indeed, by exciting optogenetically the projection of VMPO-LPS neurons to the arcuate nucleus, we see a decrease in chow consumption, absolutely no effect on body temperature. So we think this is the node that enables decrease in appetite. So these are the direct inhibitory connectivity between VMPO-LPS neurons and centers that control both temperature and appetite. In experiment that I won't have time to describe to you, we also found a number of indirect connections. So think about these neurons are inhibitory. If this projection send another level of inhibitory connection to second order neurons, the net effect will be excitatory. And so when we look at areas that are excited by VMPO-LPS activation, we found also quite a number of brain areas that are involved in temperature and appetite control. So this is summarized here. The idea is that during an infection, at the level of the CVO, we have these VMPO-LPS neurons that are activated, and through both direct and indirect connectivity, produce this sickness-mediated fever, worm-seeking, and appetite suppression. So the next question was, what's the identity of these neurons precisely? Can we identify molecular markers? And also, how are they activated? What pathway lead to these neurons being activated during an infection? So for the nature, the identity of these cells, we use single-cell sequencing. After injecting an animal with LPS, we perform single-cell sequencing of the VMPO. And we found that two clusters, this cluster 19 located here and cluster 5 located here, were CFOS positive. The cluster 5 is our non-neuronal cells. It's a mix of ependymal cells and astrocytes. And the cluster 19 is a neuronal marker, is a neuronal cluster that we were able to specifically visualize using in situ transcriptomics with my colleague Jiahui Zhuang called MERFISH, which enabled to perform transcriptomic approaches in situ. And so the VMPO LPS neurons are this blue population here. They express two markers, galanin and amigo2. And interestingly, if you look at what subset of these neurons are CFOS positive after LPS injection, which are the neurons that are labeled here in purple, you can see that interestingly, they are just at the boundary of the blood-brain barrier right here, which is obviously extremely interesting. We also look at ependymal cells and astrocytes and also found that the activated cells were either at the bottom, close to the CVO, or around the ventricle. And so this is interesting because it suggests that not only there's a specific population of VMPO neurons that is activated by LPS, but also the location of the activated cells close to the CVO and the ventricle suggests that maybe these neurons might be activated by some type of local paracrine mechanism. And so the next question was, can we find actually cytokine, chemokine, and their corresponding receptors that are expressed around the VMPO, and for the receptors, obviously, expressed by VMPO LPS neurons. And what we found was quite interesting. When we look at prostaglandin synthase, we found high expression along the blood vessel. Here in purple are the LPS-induced CFOS, so these are the VMPO LPS neurons. You can see they are very quite close from these prostaglandin synthase making cells. We found also expression of interleukin-1 beta along the ventricle, and the chemokine CCL2 also along the ventricle. So that's great. There's ability for prostaglandin, interleukin, and chemokine to act, but are the VMPO neurons expressing the right receptor? And indeed, they do. Here in green is labeled the receptor for prostaglandin EP2, and you can see the VMPO LPS neurons are both purple and green. Same for this interleukin receptor associated protein, and same for the receptor for CCL2, which is called CCR2. So these VMPO LPS neurons seem to have the right receptors to be sensitive for local cytokine, chemokine, and prostaglandin. So are they indeed affected by these signals? And so Vic Kapoor, a postdoc in the lab, performed slice recording and recorded directly from VMPO LPS neurons by adding to the slice preparation prostaglandin, CCL2, and interleukin-1 beta. And what he found is that these three molecules had a very clear effect in increasing the intrinsic excitability of the cells, as well as increasing the extrinsic excitability, so the excitatory input into those neurons, and decreasing inhibitory connectivity. He also found something quite interesting, which was there was some type of local amplification of signal with interleukin-1 beta inducing the COX-2, so the prostaglandin synthase, that in turn lead to even more prostaglandin being generated here. So overall, we found what I think is a really interesting node of sickness behavior in these VMPO LPS neurons that affect fever, warmth seeking, and appetite. Now, as I mentioned, these neurons actually project to 12 or more brain areas, and so there are likely many other effects that is triggered by the activation of these neurons during an infection. Moreover, other groups have, for example, Steve Lieberles, as well as Jeff Friedman, found also some really important centers controlling some aspect of sickness behavior in the brainstem. And so the idea is that the sickness is really a very special state that is controlled by multiple circuitry and multiple pathway throughout the brain. Now, One aspect that, however, we didn't see, and that was a little bit disappointing to us, was that the VMPO LPS neurons clearly did not affect social behavior. And obviously, this is something that we are particularly interested in the lab. And so we start to think about how to approach this. And this is what I'm going to tell you next. Now, at this point, I need to actually tell you why we start to work on sickness behavior. And that came from a meeting I had many years ago in New York, organized by the Simons Foundation. And the meeting was about a phenomenon I had never heard about called the fever effect. Now, before I go into a little bit more detail about the fever effect, I have a question for the audience. How many of you are either MDs or medical students? Can you raise your hand? None? Really? God, so all my effect is destroyed because the first time I talked about the fever effect, that was at UCSF. My host, who was Anna Malowski, who is a psychiatrist, told me she didn't believe in the fever effect. And her department chair, the chair of psychiatry, also didn't believe in the fever effect. So that gave me a pause. So I asked my MD friends about the fever effect. And then, so I'll tell you what they say. But then a little bit later, I gave a talk at Stanford, and my host was Sergio Pasca, who is also an MD. And he told me, I do believe in the fever effect because I saw it. So if there was MD in the room, your choice, but listen to what I have to say, whether it has an impact on fever effect on it. So what is the fever effect? Well, I got invited by the Simons Foundation, and they say, we're organizing a workshop on fever and autism. And I say, I think you got the wrong person because I don't know anything about fever and I'm not working on autism. They said, no, you have to come because this involves the hypothalamus and we need your expertise. So I went there, and he's Jim Simon and his wife. And this is the story he told us, that his daughter is autistic, and he and his wife noticed that each time she had a fever, her symptoms of autism disappeared for the time she had a fever. And it was so striking that he could even predict when she had a fever because her behavior changed so much. So he talked to the doctors and the scientists that were around him, and they all put him. They say, what are you talking about? No relationship, fever, whatever. So he was intrigued, and he asked other parents with children with autism. And surprisingly, many of them had similar anecdotes regarding their children. So he paid for a very large survey of parental reports on the behavior of their children when they had fever. And what came out of this is that indeed a significant portion of children, autistic children, experience a reduction of their symptoms during febrile episode, decreased hyperactivity, decreased stereotypy, improved speech, improved social interaction. So as you can see, it's not all of them. It's 1 out of 6. And maybe this is why some of the physicians are skeptical of this so-called fever effect. In any case, based on this survey, Jim Simon started again to talk to scientists and MDs, and none of them seemed interested until he talked to Emery Brown, who is an anesthesiologist, who told him, but what you say makes a lot of sense, because temperature and social behavior are both processed in the brainstem and in the hypothalamus. So maybe what's happening is that during fever, there's some local thing happening that affects social behavior and improves somehow the symptoms of autistic children. So in any case, when Jim told that story and had no idea about any controversy about this, I thought that was absolutely amazing. And that's how we start to work on sickness. And so So in this first set of neurons that we identified, there was absolutely no effect on social behavior. But then, on the side, we were starting to think, how does fever, or more generally sickness, affect social interaction? And here, Jessica Osterhout, my postdoc, made a really interesting observation. So first of all, it is very well known and published in the literature in many papers that if you put a sick and a healthy mouse together, they avoid each other. And so basically, it has been shown by Gloria Cho in particular that the healthy mouse detect pheromones out of the sick mouse, and that triggers an avoidance behavior. So that's this. But what Jessica found is that two sick mice have the opposite behavior. They have this very, very remarkable pro-social behavior where they huddle with each other for a really long period of time. And so that was very striking, and we decided, yeah, let's try to see on autism model. And we try contacting AP2 and SHANK3 mutant. I'm going to show just contacting AP2. What if you observe the time two wild type or two contacting AP2 mutant mice spend together, they are obviously less interaction with the contacting AP2 mutant, which are a model of autism. But if you inject both wild type and contacting AP2 mutant with LPS, then you see this very strong increase in social interaction and in social interaction. And as you can see, there's absolutely no difference now between the wild type and the mutant. So, maybe indeed, when animals are sick, somehow the social deficit in this contacting AP2 mice is reduced. And so how does that work? That's really what intrigued us. And so we went back to the areas that are CFOS positive after LPS injection. And the ones that are shown here in pink are the areas that are particularly involved in social behavior. And you can see that nicely CFOS positive. And one in particular was very intriguing, because that's the paraventricular hypothalamus, or PVN, or PVH, and that is the area that has the neurons that express the so-called social peptides, oxytocin, vasopressin, etc. And so we were very intrigued by this PVH activation after LPS injection, and wonder what specifically are the neural types that are activated. And amazingly, what we found is that the cells that are CFOS positive strongly express the neuropeptide oxytocin, which is a well-known prosocial peptide. And when we wonder how are these neurons activated after LPS injection, Jessica made an amazing observation, which is these neurons express extremely high level of the prostaglandin receptor EP4 that you can see here. Oxytocin is in red, EP4 is in green. You can see this perfect match between oxytocin and EP4 receptor expression. And in fact, amazingly, the PVN and these neurons are the neurons in the entire brain that express the high level of EP4. In other words, these neurons are literally poised to respond to an inflammation, a prostaglandin signal. So obviously, we need to find whether there is a causal effect between the activation of these neurons, the expression of EP4, and the prosocial behavior we observe, this huddling behavior. So what we decide to do is to specifically delete the EP4 out of neurons expressing oxytocin by crossing the oxytocin receptor CRE to an EP4 flux mouse line. And what we found is that we completely abolish huddling behavior between two sick mice. So indeed, it seems that these neurons are responsible for or are involved in this LPS-induced huddle. Importantly, we see no change in temperature preference, which means that this is not a temperature thing. It really is a prosocial behavior. We also injected oxytocin receptor antagonist and abolished huddle. And then we decided to do a gain of function, which is we took these neurons, remove EP4. So these neurons now are no longer able to to respond to LPS through prostaglandin and release of oxytocin, but now express in this nuance an excitatory dread, and look at what's happening in saline compared to LPS injection. And we found is that indeed injecting LPS and activating this oxytocinergic nuance rescued the hurdle. And these suggest that now these oxytocinergic nuance are both necessary and sufficient for the hurdle behavior that we observe. Now, this is extremely interesting in the context of autism because it has been shown in common monogenetic ASD mouse model that there is a defect in either the number of neurons that express oxytocin or the level of oxytocin expressed by some neurons, both in contacting AP2 and SHANK3B models. But also some meta-analysis in human patients show that I'm sorry, this is very tiny, but in children, but not in adult, there's reduction of oxytocin levels in patients with autism. And so what might be happening then, in fact, is that in ASD model, mouse model, and human, there are fewer oxytocinergic neurons, or there is less circulating oxytocinergic neurons. And so the phenotype is partially rescued by this sickness-induced release of oxytocin, the so-called fever effect. But there are two problems in what I just told you. One is, as I mentioned, the fever effect doesn't seem that terribly robust, and indeed is even questioned by some people. And these might actually be also associated with another issue with what I just told you, which is that, fine, so you have two sick mice that are huddling because there's oxytocin released, but what's happening when you have a sick mouse and a healthy mouse? Oxytocin is also released. And so why are these two sick and healthy mouse not huddling? You know, what is the mechanism by which this avoidance behavior is created? And I should mention at this point, actually, that when you put a healthy and a sick mouse together, they each avoid the other, which is the healthy mouse avoids the sick mouse. That was clearly demonstrated. But if we look at the behavior of the sick mouse also avoids the healthy mouse. And so, you know, the healthy mouse probably see the sick mouse as a danger, you know, doesn't want to get contaminated, but the sick mouse probably sees the healthy mouse as a danger, as a threat, as a bully, and therefore avoids this as well. So, but how is this avoidance created if we have this oxytocin release? And this is the work of Zuri Sullivan, a postdoc in my lab who was trained as an immunologist and joined the lab and did the work I will present now. So what Zuri found is something extremely interesting, which is extremely high expression of the oxytocin receptor in an area we had never heard before called the paraventricular thalamus, or PVT. You can see here, this area is both activated during sickness, is CFOS positive, and express a lot of oxytocin receptor. Now, the PVT is a non-projection site from neurons expressing oxytocin. And so we wonder, you know, what are these neurons expressing oxytocin doing in this huddling behavior? And we initially thought that they might participate to the huddling behavior. But interestingly, when we actually genetically depleted the PVT from oxytocin receptor by injecting an AAV3 in a mouse that was flocked for the oxytocin receptor, what we found is that now we increase huddling between the sick and the healthy mouse. So this is the lack of huddling when there's a healthy and a sick mouse together. But in case of the PVT no longer express oxytocin receptor, we see this increase in huddling behavior, suggesting that the PVT has a role in inhibiting huddling upon stimulation by oxytocin. So how could that work? Well, there are different populations of PVT neurons. We are here in the posterior PVT, and here are two molecularly defined populations of neurons that were identified by other group, one set express galanin and is responsible for arousal, and another subset of neurons expressing the DRD2 receptor, dopamine receptor D2, and these neurons lead to aversive behavior. So that fits perfectly. And interestingly, when we look at the input from PVT-DRD2 neurons, we found that there are a lot of brain area that project here that are known to respond to predators or threats, for example, the PAG or the parabrachial nucleus or the BNST. So it seems to fit quite well. And indeed, I won't have time to describe this experiment to you, but when we manipulate genetically, chemogenetically, these PVT-DRD2 neurons, we found that they are indeed both necessary and sufficient to inhibit huddling behavior between a sick and a healthy mouse. So now we have a pathway that leads to huddling and a pathway that inhibit huddling. And we also, by doing photometry in the PVT, we found that there is indeed an increase in activity after LPS injection, and this activity is completely inhibited by oxytocin receptor antagonists. So indeed, these neurons are activated during sickness and in the presence of oxytocin. So how does that work then? So the idea is that prostaglandin activate oxytocinergic neurons, and this has two effects. One is huddling behavior when two sick mice are together, and the other one is avoidance in a different context, which is when a sick mouse encounters a healthy mouse. So the idea is that according to the context, if this is a friendly environment, then the prosocial pathway that was identified by Rob Malenka, Golendul, etc., leads to prosocial behavior, so huddling, but in a context in which there is some type of threat, then it's a different pathway that is activated. That's the PVT. And I think there's quite a number of interesting findings here. One is that these neurons expressing oxytocin really are at the interface between the immune system and social behavior segment. And they can act in two opposite ways, pro-social or anti-social, according to the context. And I think this, for me, is super interesting in the context of the debate about fever effect, because maybe what's happening is that the fever effect works in children that are relaxed and comfortable, and on a child, autistic child that is freaking out, it has exactly the opposite effect. And I think this is interesting not only in the context of the fever effect, but also in the context that, you know, people have actually tried to administer oxytocin in autistic children, and it doesn't work. Well, maybe it works sometimes. Maybe it works on children like this and doesn't work on children like this, because it's exactly the opposite effect that is generated. Now, you know, when I was telling this to some of my European friends, European MDs, they say, hey, there's nothing new here. And this reminded them of the old style pyrotherapy. So you may know or not that the first Nobel Prize in psychiatry was given in 1927 to a psychiatrist for malaria therapy for general paralysis of the insane. What this was is that that gentleman discovered that if he injected malaria or tuberculosis of a number of nasty stuff to patients with psychosis, many of them felt better. In fact, many of them were able to leave the asylum at that time because it has this amazing effect. Now, obviously, this effect was very immediate. And, you know, some of them had longer term effect, like, you know, getting malaria or tuberculosis. And so the pyrotherapy actually lasted for quite a while, but then was supplanted by electrotherapy. So it disappeared out of the repertoire, but still is there in memory. And in fact, there is this very interesting quote for Parmenides around 500 BC, give me the power to produce fever and I'll cure all diseases. So wonderful, wonderful quote, I think. So anyway, I think this really forces us to view this interaction between the immune system and the prosocial behavior circuit and maybe this fever effect in a different way. So, so what I've shown you so far is these two nodes of infection and sickness behavior. But the idea is that this might only be the tip of the iceberg, which is that to start with, in a paper that we published in collaboration with my colleague Jiawei Zhuang on the aging brain, we found that there are a lot of gene expression changes in the aging brain, and the majority of these changes are shared with LPS-induced brain inflammation. And so we realized this because when we look at gene expression in aging versus adult brain, many of the genes that had their expression change were related to inflammation. And we tested that directly by now injecting LPS to a mouse and looking at gene expression differences. And as you can see here, these dots, these dark dots here, are all the genes in all these different cell types that are common between LPS and aging brain. And so we tested this idea specifically, and indeed we found that old mice hurdle way more, both normally but also after LPS injection, suggesting that there are different forms of inflammatory condition that affect behavior. So now let me switch to the last part of the talk, which is social isolation and prosocial behavior. So I'm showing these two paintings because for me, I love art. And for me, it's very striking that each of these paintings reflect very strong emotion. And the emotion reflected in one painting is the opposite than the other. Here you have people together, they're having fun, there's joy in the air, people interact with each other, are happy. This is a lonely person, and the painting is there to reflect sadness. And so you might wonder, why is that? Well, it's a very trivial explanation, which is, you know, when you grow up, you learn that being with your friends is fun and being alone is boring. And so this is happy and this is less happy. But it turns out that more generally, there is something very positive about being together and something very negative about being alone. And this can be seen by looking at the consequences of social isolation. So people who are isolated have increased stress, fragmented sleep, a weakened immune system, increased cardiovascular risk, impaired cognition, and the list goes on and on. In fact, there was a report by the US Surgeon General last year in 2023 that talked about our epidemic of loneliness and isolation and called the loneliness an epidemic and a very strong health risk. And interestingly, shows that's good for the smokers, I guess, but that lacking social connection is as dangerous as smoking up to 15 cigarettes a day. I mean, that's, you know, pretty impressive, I have to say. And so it's not good to be alone. And by contrast, there are a lot of advantages of being together, you know, for finding a mate, to defend against predator, for thermoregulation, for doing things together, whether it's finding food, parenting, and doing just things together. And so one idea is that being together is actually a fundamental evolutionary conserved drive. The brain is telling individual, you cannot be alone. It's bad for you, and you have to be with others. And this idea of this drive to be together has been studied in early work by Panskip, Cacioppo, David Anderson, Ketai, etc. And we started to work on this a number of years ago with a very particular angle to it. And the angle is the following, which is that if you observe mice that are together in a cage, so cage mate, and you take one of these mouse and you put it alone in a cage for a few days, and then you bring it back together with the former cage mate, there is an amazing increase in social interaction that occurs coming from the formerly isolated mouse. And this is what's shown here. So here's 0 isolation, 1 day, 3 days, 5 days of isolation. And each of these blue tick here represent a bout of social interaction. As you can see, the longer the animal is isolated, the more it interacts with the other. So it has a rebound in social interaction. And this rebound is extremely interesting because it reveals a fundamental homeostatic need. What do I mean by this? Well, we all need to drink, to eat, and to sleep. And when we are water, food, or sleep deprived, it triggers in us a need state. And if this need state trigger motivated behavior, you want to drink, you want to eat, you want to sleep. And if this need is not restored right away, then it leads to a rebound, which is that the more water deprived, the longer water deprived you are, the more you drink, the more food deprived you are, the more you eat, the more sleep deprived you are, the longer you sleep. And so what that means is that somewhere in the brain, there's a bean counter of what you drink, what you eat, how much you sleep, and that bean counter is driving that need, and the needs becomes more and more elevated. The bean counter becomes more and more active as the need keeps increasing. And so we think that similarly, they could be also, based on this rebound, some type of bean counter of social interaction that drives the animal to seek others when it's alone. Now, interestingly, these needs have been modeled with this increase in need and then this period of satiation when you sleep, when you eat, or when you drink. And that's the idea, is that there's something similar happening when the animal is alone. Now, what is interesting is that in all these cases, sleep, drinking, and eating, it's distinct hypothalamic population that have been identified that play a key role in physiological homeostasis. So similarly, we think maybe the hypothalamus is also the place of the bean counter for social drive. And moreover, in the case of hunger, for example, there are these really interesting neuronal population that have been identified. For example, AGRP neurons in the arcuate nucleus that drive hunger, and arcuate POMC neurons that drive food satiety. So you have these neurons that have opposite function, one driving the need and stimulating the need, and the other one encoding the satiety of that need. And so similarly, we wonder whether there could be in the hypothalamus these two populations of neurons, one responding to social isolation and driving the social need, and another one activating during reunion and driving social satiety. So we decided to look for these neurons. And for these, we first look at different mouse lines. As you can see, these are 57 black 6. These are the typical mouse line that people use. As you isolate the animal for longer days, you can see this increase in social interaction, this rebound. But interestingly, we found that there are a lot of mouse lines that actually have very little, almost no rebound. So they are completely insensitive or almost completely insensitive to social isolation. And there are mouse lines that have exactly the opposite, that are extremely sensitive to social isolation, have this extremely robust rebound, in particular this FVB line that you see here. So all the studies I'm going to tell you about have been done both on C57 and FVB mice. Oh, and I should say, all these studies have also been done in female mice, because if you male mice, when they are isolated and then brought back together, have all this aggressive behavior, and if they are reunited with a female, they will mate. So, to avoid all this competing drive, social drive, we have been using just adult females. But the data I'm going to show you, we also found in males. And I'm not going to show any of this data, but I can respond to questions if you're interested. So the idea, the model we have is that in a similar way as the hunger neuron, the AGRP neurons in the arcuate nucleus that start firing as soon as food deprivation starts, and then stop firing as soon as the animal access food. And you have these food satiety neurons that start to fire, the POMC neurons, as soon as the animal has access to food and up to the end of the reaching the satiety. We also hypothesize that there might be neurons that start to be activated when the animal is isolated, stop their firing when animals are reunited, and at that time you have another population of neurons that are encoding the grouping and the social society. And so we decided to image using microendoscopy this area of the hypothalamus called the MPN, or medial preoptic nucleus, because that's an area that contains a lot of neurons involved in social interaction, in social behavior in general. And so here's what we found. So this is a mouse that has this microendoscope. And in the first part here, this mouse has been isolated for 3 days. And in the first part here, you will see the firing of neurons in this isolated mouse. And then we will put a former cage mate here, and you will see the change of firing of these neurons when the animal is reunited. And so here's this mouse by itself. And you can see we put in different color here the firing of two populations of neurons, this yellow one that have very low firing and this green one that fire quite strongly. Now we part, and so you see this rebound here, the mouse say, I want more interacting with you, very eager to interact. And as you can see now, these neurons that were quiet now start firing like crazy. And these neurons that were firing very strongly when the animal was alone now stop firing. So these perfectly correspond to our model here of these reunion neuron and isolation neurons. And so the question now is, can we identify what these neurons are molecularly and manipulate them genetically to see how they work? And so we have a number of years ago create a cell atlas, a cell type atlas of the medial preoptic nucleus. We know all the cell types, all their different markers. So by doing CFOS after social isolation or after reunion, we can then use just candidate markers, and identify precisely what is this population that is activated during isolation, what is the population activated during reunion. And we found two perfect hits. The neurons that are express VGLUT2, so they are excitatory and express the marker MC4R, shown here in green, correspond perfectly to the isolation neurons. And neurons that are VGAT positive, so inhibitory, and express the marker THRH, here in purple, correspond to deactivation during reunion. So we have these two cell types. And interestingly, we found that the reunion neurons directly project, has direct synaptic contact with the isolation neurons. And this is extremely interesting because these reunion neurons are inhibitory and the isolation neurons are excitatory. And so one hypothesis on how the system could work globally is that when animals are together, these reunion neurons are active, and therefore they inhibit the isolation neurons. So there is no social need because the social need is fulfilled, animals are together, and these isolation neurons are silent. But when the animals are now isolated, these reunion neurons no longer fire, therefore the isolation neurons start to fire and provide and encode social need that drives the animal to seek social encounters. So are the isolation and reunion neurons doing what we predict they do, using MC4R-CRE and translate MC4R-CRE and VGLUT2 flipase mice? We found that indeed when we optogenetically activated the isolation neurons, we increase social seeking. We found that this effect is long-lasting, which is we can activate these isolation neurons when the animal is alone, and then put it with another mouse without activation and still see this increase in social interaction. So this is interesting because it means the activation of the isolation neurons lead to a particular state of social seeking. And then finally, we found that if we perform a two-chamber assay in which we activate the isolation neurons in one of the chamber. When the mouse is in one of the chamber and not the other, the mouse will ultimately carefully avoid the place in which the neurons were activated, suggesting that these neurons encode an aversive state. So this is really interesting because what we show here is that these MC4 VGLU2 neurons encodes both social need and also conveys negative valence, exactly what social isolation does. And similarly, we found that the reunion neurons repress social need and convey positive valence. So it's good to be together. It's not good to be alone. How do they do that? Well, for the isolation neurons, we could find what their various projections are. And three projections for us were particularly interesting. The PVN, again, you heard about it. The social peptide are there. The lateral abenula that may convey avoidance, and the arcuate nucleus that control food consumption. And so indeed, we were able to demonstrate genetically that these isolation neurons directly project to neurons expressing oxytocin, and that by inhibiting oxytocin receptor activation using oxytocin receptor antagonists, we suppress the rebound, and we increase the rebound when we inject, when we stimulate oxytocinergic neurons. So we think that what's happening is that during social isolation neurons drive the release of oxytocin in the animal alone, and that oxytocin level increasing in the animal really drive that social seeking. And that's the state of social drive that is being generated during isolation. We also demonstrate that these isolation neurons directly project to neurons of the lateral abenula that express the marker GAP43, which is a marker of neurons generating aversive states. And when we stimulate this projection, we induce an avoidance of the chamber in which these neurons were stimulated and has no effect on social interaction itself. So it's a particular node controlling aversion. And then finally, when we have animal isolate, they lose weight, and we found that these isolation neurons actually directly project to POMC neurons. These neurons, remember, encode food satiety, and the animal just stop eating when it's isolated. So overall, and these three projections really convey already three major effects of social isolation. So the last question I want to address, and something that I was very intrigued, is how does an animal know that it's together or alone? Is it because it sees the other one, it hears, it smells the other one? What is it? What is the information that the animal captures that tells it, I'm alone or I'm together? And so when we isolate animal, when we isolate them entirely, obviously they no longer have any sensory information about others. There's nobody around. So what we did instead is using these perforated dividers in which they can see each other, hear each other, smell each other. And interestingly, when we do this, they still have a social rebound, meaning that none of this modality, hearing, seeing, smelling, none of this is sufficient to tell the animal they are not alone. They still feel alone, even through that divider. And so what's left are modalities that involve contact. And there are two of them that I know of. One is pheromone detection in mammals, required touch. And we could quickly eliminate this because when we take mice that are deficient in pheromone sensing, they have a normal social rebound. So pheromone do not play a role here. So the last one is touch. And so does touch plays a role? Well, there's been a number of sensory neurons that have been hypothesized to be involved in social touch. One are the C low threshold mechanosensory receptors, a very large category that are likely to include neurons involved in social touch and can be recognized with the marker NaV1.8. And then this marker MRGB4 that was studied by David Anderson to start with, and Ismail Abdou-Sabor more recently. And so we decided to look at mice in which either of these population was ablated using genetic tools and DTA cell ablation. And what we found is that in each of these cases, we indeed reduce the rebound, suggesting that touch is the one modality that is essential for animals to know they are together or alone. And I personally, we found this right coming out of the pandemic. And I thought that was so amazing because we do know the role of touch in social interaction. When people were sick with COVID and they had one of their family member in hospital, they couldn't even hug each other, they couldn't touch each other. It was absolutely horrifying. So comfort touch really has this amazing role socially, and indeed might be the signal that conveys that animals are or individuals are alone or are together. Now, my postdoc, Ding Lu, who did all this work, came with a very ingenious idea. And that was inspired by a very famous experiment, the one done by Harlow in the 50s, in which young monkeys were given a choice between what he called a wire mother, that, as you can see, had all wire but food and drinks, and this closed mother that had this very soft fabric but no food or drink. And very systematically, the young monkey chose the closed mother, so chose the soft touch rather than the food and drink, telling how important that comfort touch could be. And so Dean came triumphantly to my office one day and say, I was able to design a Harlow experiment for mice. And that is this, which is that he put this soft fabric in a tunnel. Mice love to go into tunnel. So he took this plastic tunnel and put this soft cloth in there. And mice love that little closed tunnel. And so he tested the influence of this soft touch, or whether the mouse had a preference for the plastic or the closed tunnel. And he found something very interesting, which is when the animals are together and you give them a choice, they don't care. They go to either one. But when the animal is isolated, it goes to the closed tunnel preferentially. Moreover, if you isolate a mouse and you put the isolated mouse either with the plastic tunnel or the closed tunnel, and then reunite it and check the size of the rebound, you can see that when the animal has this soft touch together, it has way less rebound than being in the plastic tunnel, suggesting that indeed soft touch during social isolation significantly reduces social need. And why is that? Well, he's the version 2.0 of the closed tunnel. You see it's now very even more comfy, has this. And then it has an opening here, so we can actually have a mouse that has a micro endoscope on top of its head. And here's what's happening. Here's the mouse with the micro endoscope, and it loves going into the little tunnel. It goes there, goes again and again and again. But here, the advantage is that we can image the neurons when the animal does that. And so here's the paradigm. We isolate the mouse. We have the mouse, playing with the tunnel, and then we reunite the animals. And when animals are reunited, we can identify the neurons that stop firing, these are the isolation neurons, and the neurons that increase their firing, these are the social society neurons. And now we can check what is the activity of these neurons when the mouse crosses the tunnel. And what we found is that the reunion neuron, the society neurons, fire each time the mouse is crossing the tunnel, here, here, and here, and here. And the social isolation neurons stop firing each time the mouse is crossing the tunnel. So it fits really quite well with this idea of soft touch being important for social society and detection of needs. So here are the model we have. We have these social society neurons that we've molecularly identified projecting to social needs neuron. These neurons are activated by the presence or the absence of touch, and they lead to an array of behavioral changes of social seeking, negative valence physiological changes by virtue of very specific projection, and similarly for social society that leads to reward and social society. So what's next? Well, we don't know how any physiological needs accumulate, but social needs certainly seem something very interesting. We also know that animals or individuals within a species or cross species or cross ages have very different social needs, and what is the origin of these differences is something very interesting. We all know during the pandemic, some people were absolutely distressed to be alone, and some people were super happy to be alone. We've seen with these different mouse strains that some are happy, some don't care, and some are distressed. Anyway, these are finding these three population of neurons, which we think are very likely to interact with each other. They are all more or less in the same place in the brain. And in fact, all these needs are likely, and behavior are likely to interact with each other. We're very interested in identifying this matrix of cellular neuronal interaction, and also the development of this circuitry. And on this, let me thank the people who've done the work. Jessica Osterhout is the one who first started the sickness project that has now been taken over by Zoe Sullivan and Vic Kapoor in the lab, with the help of a number of members of the lab. Social homeostasis was done by Ding Lu and Mostafizur Rahman, and here are our collaborators and funding sources. Thank you.
Thank you. Time for questions, if there are any from the audience. Students, there are any students?
It's an interesting question. It was detected on individuals that have a defect in social interaction that suddenly seem to have this improvement. In principle, there's no reason to believe this would not exist overall, but you might just not see it because, you know, non-autistic individuals don't have a social defect, and therefore you might not see a difference. In that case, you know, you overcome a deficit, which is easier to detect.
The moment there's fever, the person feels that they need help, and therefore they are more invited to have a social interaction.
Yeah, and indeed, that is probably the case, right? So when you have an infant that feels sick, they might indeed need to be comforted by someone. So that would fit also perfectly. I think that, again, you know, there is this just really interesting observation in the case of a particular disorder where it might be a compensation. And in particularly in the case of autism, because the autism was associated in many circumstances with a defect in oxytocin level. So, you know, it's just a very simple model that you just, improve the autism by just relieving the deficit in oxytocin. But again, you know, this seems way more controversial than I had anticipated. And I just think that some of the controversy comes from the fact that, you know, 1 in 6 individuals is not a lot. It seems very robust in the sense that it's very reproducible, but 1 in 6, you know, it's not 40%, it's 1 in 6, it's not a lot. And, you know, when I talk to MDs, they tell me that they just worry that, you know, parents association, like parents of children with autism, are extremely eager, rightfully so, to find ways to help their children. And I think physicians are a little worried that suddenly there is, you know, some snake oil that is, you know, being promoted, and that, you know, they wouldn't be able to do anything about it. I mean, part of it is, I think, also, frankly, that physicians tend not to believe too much the parents, when in fact, you know, I think the parents are probably the best at observing the behavior of their children. I mean, again, I found it very striking, the dichotomy between people who say, yes, I believe because I saw it, and people who say, I don't believe it.
I understand well why the isolation experiments with males and then put back to the patient. But I do wonder, are there sex differences in these neurons in the way that they change them as lack of capacity? Have you looked at males in younger groups for huddling with LPS when they're not aggressive, or do they respond to a something?
Okay, so you're talking about two things. You're talking about sickness and you're talking about social isolation. In the case of sickness, the initial project was done exclusively in males because females have fluctuating temperature according to the estrous cycle. And so that really would provide a lot of difficulty. But that's the initial part. Then the LPS hurdle, we found this both in males and females. There's no difference. It is very robust in both cases. Relate to social isolation, it's interesting that you asked the question because in fact, we've looked at, we've done single cell sequencing of hypothalamic development, and there are two cell types that are the most sexually dimorphic cell types in terms of difference in gene expression. And it's the isolation neurons and the social society neurons. And we don't completely know what to make out of it, but we are working on it. That's what I can say. So males have a rebound, but way less. And we also see that there are all these differences in gene expression and how does it fit with it? Are there some cell intrinsic gene differences that explain the difference in behavior? That's something we're very interested in observing. Yep.
Thank you so much for your great talk. My question is about the neurons and their role in immune-related and social behaviors. As I don't know if they are like different models of disease, but I saw that there are lots of like LPS and inflammatory agents to like some specific disease models. I wanted to know how these results can be generalized. So like, and also can the consequence be like changed in different conditions like autoimmune conditions? And also I wanted to know if there is a possibility that maybe the oxytocin neurons have like the effect of buffering social behaviors during illness so that it can help for recovery and
yeah. So let me start by your second question. I don't know anything about buffering. We've not tested that. I think it's a good idea. We just have not looked at this. Relate to different inflammatory conditions. I have a grant due next week, and that's exactly on that topic, which is that indeed we think that different inflammatory conditions have very different effect on social behavior. So for example, LPS trigger huddling, aging triggers huddling. There are few other compounds that trigger huddling, but there are a number of inflammatory conditions that do not trigger huddling at all, but have an effect on the social homeostasis circuit, which LPS does not have any effect. So, so all of this, for us is super interesting. I have to say, when Zuri Sullivan, who is again an immunologist, when she joined my lab, she told me that she expected that different inflammatory challenges would create different sickness behaviors. And I told her I was a little skeptical. As an immunologist, she's used to the idea of the specificity in the response. That's what the immune system does. But from a behavior standpoint, I couldn't really figure out why you would need some type of specificity. And it reminded me of something that Charles Zucker say about taste receptors. Sweet is good, because it's full of calories. And it turns out that bitter compounds that are repulsive tend to be toxic agents. And so the way the taste system works is that, the sweet receptor tests any type of sweet, tells you it's sweet. And then you have multiple beta receptors because beta substances can be structurally very different, but all the beta receptors are expressed by the same taste cells. And the way he says is, you need to know it's good or bad. You don't need to know the specifics of the good or the specifics of the bad. And so for me, I saw inflammation in exactly that way. It's inflammation, you have sickness behavior, but Zuri thought that was not the case. And I think she's right. To what extent, and can we find also some type of logic to what a substance does or inflammation does or not, still to be defined. I hope the reviewers of my grant will like it.
Where you record the endurance, so the reunion or the isolation endurance in the MDM, when you introduce the new mouse today, so it looks like that you show that the new mouse kind of avoids the one that's already created. So I was wondering if you ever recorded isolation or reunion endurance in the new mouse.
In the other mouse.
Yeah,
we did, and we are currently analyzing the data, and I don't know what the answer is, but yeah, we thought about it. I think one of the problems is that the isolated mouse is so eager that it kind of freaks out the other one a little bit. I'm sorry, I'm having trouble to distinguish.
Consider looking at factors like CRF, for example, because there are other projections that go to the same area from the BT, for example.
Yeah.
Whether you expect some sort of like feedback or more like an amplification or anything like that.
Yeah, we are starting to look into this. What we find is that it seems that EP4 is specifically expressed by osteotocinergic neurons, not the other population, but that after LPS injection, there are some AVP neurons and some CRH neurons that are activated. So they are probably part of what's happening, but we don't know where they play a role.
Way in the back.
sure. Wonderful talk, thank you. With regards to the part of your talk, this is more a question about the experiment itself. We looked at the aversion region of PDT, I believe, and when you inhibited it, you saw in a healthy sick interaction, you would increase the interaction time. Now you mentioned that the sick animal is probably releasing some sort of pheromones or behaviors that are keeping the healthy animal from interacting, as if that healthy animal doesn't want to be around there. So when you're inhibiting, you're just inhibiting the sick animal, and so that is also responsible for the secretion of these pheromones or these targets. That's telling the healthy animal like stay away from it.
Yeah, but they still huddle. They huddle more than in the non-treated condition. Yeah.
The healthy pursuing the healthy animal pursuing the sick animal more so than
The healthy animal is not treated. I understand your question. I mean, in principle, the healthy animal should not change, should still avoid the sick mouse. But if one is eager to huddle, I guess they end up huddling. I don't know how to explain it,
Actually, asked the last question, which was one of the neurons that you turned up that identified touch was actually an NAD1.8 positive neuron, which is normally I think thought of as a nociceptor. So
yeah. So I might not have been eloquent enough when I describe it. It was to label the touch neurons using a marker that includes them. It's not specific of touch neuron. Absolutely. Yeah, I'm aware of that. And we tried other markers and for some reason the mice didn't breed or we had some issues. Yeah, but absolutely. You're absolutely right.
But they were seeking out gain.
No.
All right. So thank you so much.
You're very welcome.